Mendels Laws. Based on his experiments of monohybrid grass and dihybrid grass, Mendel proposed three important laws which are now called as Mendel's laws of heredity. Law of dominance: when two homogeneous, that is homozygous, individuals with one or more sets of contrasting characters are crossed, the characters that appear in the F1 hybrid are dominant, and those that do not appear in F1 are recessive characters. Law of segregation or law of purity of gametes: when a pair of contrasting factors or genes or allelomorphs are brought together in a heterozygote or hybrid, the two members of the allelic pair remain together without mixing, and when gametes are formed, the two separate out so that only one enters each gamete. Law of independent assortment: in case of inheritance of two or more pairs of characters simultaneously, the factors of gene of one pair assort out independently of the other pair. Chromosomes, DNA, and genes. The human body is made up of million cells. The nucleus of each cell contains thin thread-like structures called chromosomes. The term chromosomes was first coined by Valdier in 1888. The chromosomes are the carrier of genetic material which contain the heredity information. The chromosomes are highly condensed coiled chromatin fibers packed with the DNA, deoxyribonucleic acid, that forms the genetic material. Genes are segments of DNA which are responsible for the inheritance of a particular phenotypic characters. Each gene is present at a specific position on a chromosome called its locus. During cell division, the genetic information present in the genes are passed from one generation to another. Structure of a chromosome. The chromosomes are thin, long, and thread-like structures consisting of two identical strands called sister chromatids. They are held together by the centromere. Each chromatid is made up of spirally coiled thin structure called chromonema. The chromonema has number of bead-like structures along its length which are called chromomeres. The chromosomes are made up of DNA, RNA, chromosomal proteins, and certain metallic ions. These proteins provide structural support to the chromosome. A chromosome consists of the following regions: primary constriction, secondary constriction, telomere, and satellite. Types of chromosomes based on the position of centromere. Based on the position of centromere, the chromosomes are classified as telocentric, acrocentric, submetacentric, and metacentric. Telocentric: the centromere is found on the proximal end. They are rod-shaped chromosomes. Acrocentric: the centromere is found at the one end with a short arm and a long arm. They are also rod-shaped chromosomes. Submetacentric: the centromere is found near the center of the chromosome. Metacentric: the centromere occurs in center of the chromosome and form two equal arms. They are V-shaped chromosome. Submetacentric: L-shaped chromosomes. Types of chromosome based on its function. The eukaryotic chromosomes are classified into autosomes and allosomes. Autosomes contain genes that determine the somatic body characters. Male and female have equal number of autosomes. Allosomes are chromosomes which are responsible for determining the sex of an individual. They are also called as sex chromosomes or heterochromosomes. There are two types of sex chromosome, X and Y chromosomes. Human male have one X chromosome and one Y chromosome, and human male have two X chromosomes. Watson and Crick model of DNA. DNA molecule consists of two polynucleotide chains. These chains form a double helix structure with the two strands which run anti-parallel to one another. Nitrogenous bases in the center are linked to sugar phosphate units which form the backbone of the DNA. Pairing between the nitrogenous bases is very specific and is always between purine and pyrimidine, linked by hydrogen bonds. Adenine A links thymine T with two hydrogen bonds A, two hydrogen bond T. Cytosine C links guanine G with three hydrogen bonds. This is called complementary base pairing. Hydrogen bonds between the nitrogenous bases make the DNA molecule stable. Each turn of the double helix is 3.4 nanometer. There are 10 base pairs in a complete turn. The nucleotides in a helix are joined together by phosphodiester bonds. DNA replication. DNA replication is one of the basic process that occurs within a cell. DNA molecule produces exact copies of its own structure during replication process. The two strands of a DNA molecule have complementary base pairs. The nucleotides of each strand provide the information needed to produce its new strand. The two resulting daughter cells contain exactly the same genetic information as the parent cells. DNA replication involves the following steps: origin of replication, unwinding of DNA molecule, formation of RNA primer, synthesis of new complementary strand from the parent strand. Significance of DNA. It is responsible for the transmission of hereditary information from one generation to next generation. It contains information required for the formation of proteins. It controls the developmental process and life activities of an organism. Sex determination. The formation of zygote into male or female sex during development is called sex determination. Sex is determined by the chromosomes of an individual. Sex determination in human. Recall that human beings have 23 pairs of chromosomes, out of which 22 pairs are autosomes and one pair, that is 23rd pair, is the sex chromosome. The female gametes or the eggs formed are similar in their chromosome type, 22 plus XX. Therefore, human females are homogametic. The male gametes or sperms produced are of two types. They are produced in equal proportion. The sperm bearing 22 plus X chromosomes and the sperm bearing 22 plus Y chromosomes. The human males are called heterogametic. It's a chance of probability as to which category of sperm fuses with the egg. If the egg S, that is X, is fused by the X bearing sperm and double X, XX individual female is produced. If the egg X is fused by the Y bearing sperm and XY individual male is produced. The sperm produced by the father determines the sex of the child. The mother is not responsible in determining the sex of the child.